In this protocol, we not only provide a detailed explanation of each step of EDU incorporation and immunostaining, but also explain image acquisition, image processing, and provide some tips for analyzing large datasets. This technique allows for the allocation of cells in G1, S, and M phases. This would be particularly useful for cell cycle analysis on studies on null mutations affecting mitotic genes. We would expect people to struggle for 2 to 3 months if they do not have prior knowledge or experience with this technique.
To begin, add Schneider's dissection medium, or SM, to 2 consecutive depressions of a glass spot plate and add PBS to the subsequent depression. Using a pair of forceps, pick wandering third instar larvae and place them on a tissue wetted with PBS to clean off fly food residue. Place the larva in the depression with PBS, followed by the consecutive depression containing SM. Next, under a dissection microscope, use fine forceps to remove 3/4 of the lower body of the larva. Then gently grab the larval mouth hooks with one pair of forceps and hold the cuticle at the other end with the other pair of forceps. Turn the larval head inside out by pushing the mouth hooks inward and simultaneously peeling off the tissue at the other end. Observe the larval brain with attached imaginal discs. Remove other tissues attached to the brain and transfer the brain to the subsequent consecutive depression with SM.